So good morning, everyone. Thanks for coming. I'm Sungjoon Park, and I'm currently a postdoc at UC San Diego. I work with Dr. Trey Eidecker, and I'm so happy to be here, present my work at ISMB, which I've always dreamed of. And the story that I would like to share today is about basically a deep learning model for precision medicine. And at this point, I think the key question is not whether the deep learning will impact oncology. That is already clear, but it is how it will do so and what developments are needed to realize its full potential. And today, I'd like to engage your interest in our recent endeavor to address that challenge, which, as you can see in the screen, by a deep learning model of tumor cell architecture that explains the response to a therapy that targets CDK4/6 proteins. So needless to say, deep learning has transformed the way we understand, the way we treat disease. However, in most cases, from a biological point of view, not in all cases, but in most cases, their black box nature often hinders deeper mechanistic understanding of underlying biological processes and functions. And the pioneers in the lab, some of whom are sitting right here in this room, have established a framework called visible machine learning, where they open the black box and they actually shine a light on it by attaching the hidden layers to known biological components such as genes and pathway and broad processes such as organelles like mitochondria and nucleus and ultimately to a cell. And this knowledge map takes a form of hierarchy, and the other pioneers in the lab has built the hierarchy of protein assemblies in cancer by generating and analyzing the protein-protein interaction measured by APMS technique, integrating with massive publicly available multi-omics data that informs protein-protein information. So the part of the hierarchy can be explained by the known cancer biology, such as cell cycle, extracellular matrix, or immune system, but also they newly link systems like collagen complexes or actomyosin complex to the cancer progression. So this work that I'm presenting today is basically a synthesis of these two advances or two approach. One is cancer cell map and one is visible neural network. So what we did was we actually embed this cancer cell map into a neural network and translate the genotypes of cancer tumor samples into the phenotypes to understand the drug response. So once it was trained, it was not only able to predict the drug response, but also it provided mechanistic insights by decoding the genetic trace from genes to disrupted pathway to large protein complexes. We use GDSC and CTRP to train our model, which are the widely known cancer drug response data sets. And we were able to collect around 1,600 cell lines that were exposed to CDK4/6 inhibitor called palbociclib, and which were labeled with and annotated with the drug response represented as area under drug response curve, or AUC. And the motivation behind focusing the CDK4/6 inhibitor in the study was based on that the CDK4/6 inhibitors such as palbociclib, ribociclib, and abemaciclib, shown in this top blue box, they kind of revolutionized the breast cancer therapy as they were designed to selectively target the CDK4/6, which are the key players in cell cycle processes and one of the very known hallmarks of cancer. But people immediately face the challenge that half of the patient will develop a resistant mechanism due to the mechanisms that were largely unknown that really strongly motivated us to uncover the drug-resistant mechanisms that systematic way and comprehensive way using our approach. And I'll just quickly go through this slide about the predictive performance that our model was able to accurately distinguish between the actual resistant tumors, actual sensitive samples and actual resistant samples, not only in cell lines, but also in PDX and patients. But here is the highlight of our work that I like to demonstrate here is that we were able to systematically understand this intricate and complex drug response on top of the hierarchical map of cancer protein assemblies that was embedded in the neural network. So we compute important score for each and every protein systems in the hierarchy. So we identify 33 important systems that were governing the drug response, and we further narrow it down to 8 core assemblies, which are colored red here, that passed another additional statistical significant test and with the non-redundancy test. So for example, let's digest a little bit. So the model was able to identify CDK holoenzyme complex 1, which contains a CDK4 and 6 and RB1, which are the direct targets of palbociclib and the downstream of the direct targets, which was good to see because it can serve as positive control. But we also wanted to systematically validate our core assemblies, whether they're actually important for palbociclib response. So we were able to find out a genome-wide CRISPR knockout screen followed by the palbociclib treatments. And what we observed that what the hits from the screen, which means the genes when it knocked out strongly modulates the palbociclib response, were actually more enriched in the core assemblies than the assemblies that were defined as non-important by our model. And furthermore, we also pinpointed one assembly that was not previously appreciated in the context of palbociclib, which was a histone-mediated transcription regulation assembly that contains the genes listed here. And the wonderful lab crew in the lab had carried out this CRISPR-A experiments where they overexpress gene in this complex and then see whether the overexpression actually lead to drug resistance. So using the EDO assay that trace the cells entering the S phase, that is indicator of cell cycle, and we could actually see through a microscope that when we overexpress TBL1, XR1, and KAT6A, we actually saw that it made the cells more tolerant to drug exposure. So we actually validated one of the hypotheses that was generated from our visible AI model. And I think I could wrap up here that I show just a demonstration of AI that can guide mechanistic findings in the drug response application. But I shared the part of this. I only shared the part of the story. If there's anyone who are interested in the full story, please go check out this paper, which was published earlier in Nature Cancer. And I cannot thank enough all the people listed up here, but especially my advisor, Dr. Trey Heidecker, who I always look up to, and Erica Silva and Akshay Singhar, who are the first author of the paper. And thanks again, and happy to take any questions.
Thank you. Questions?
Hi, great talk. I would be interested in how were you able to get like the interpretations or some sort of like explanations in the visible neural networks that you created?
Yeah, so that's a good question. So we actually use a surrogate linear model to once we train our model, we take all this like hidden neurons from that was predicted by our model, and then we kind of train another surrogate linear model to see whether this hidden prediction can actually were predicted for the prediction made by this entire model. So we kind of relied on the surrogate model to get the importance score.
Thank you.